Hi everyone, my name's Amy Jackson and I'm from the University of Southampton, the Natural History Museum. I'll be talking to you today about different phylogenetic techniques and the hybridisation and ecological speciation of a Canary Island plant group called Descurainia. The Canary Islands is a volcanic oceanic archipelago made up of 7 islands which are found 100 kilometres off the west of Africa. All islands were formed within the past 24 million years and have a dynamic geological landscape. The climate here is strongly influenced by the northeast trade winds. This humid air, in combination with the dynamic topography, produces markedly different ecological zones. Here's a rough map of the ecological zones in 4 of the islands. If we take Tenerife, for example, Tenerife is around 60 miles in length, and within that there are at least 5 different ecological habitats. And so this dynamic landscape of oceanic islands, along with isolation, has resulted in high levels of endemism within Canary Islands. So there are around about 570 endemic plants here. And while insular organisms have long been used to study evolution, there are still many problems and gaps in our knowledge. Phylogenetics is a key part of framing our hypotheses. However, many Canary Island phylogenetic studies have failed to resolve relationships within these groups. And following this, often evolutionary processes such as geography, ecology, and hybridisation are difficult to identify. And this is mostly due to the lack of genetic and genomic data available. And many studies have used low copy chloroplasts or nuclear markers that are unable to fully resolve relationships. But with the advent of next generation sequencing, we can provide a more comprehensive look into island plant evolution. So my study system is a genus called Descurainia, which is in the mustard family Brassicaceae. There are 45 species in total within this genus, but I look at the 7 endemic species to the Canary Islands. And Descurainia is often characterised by the long erect stems and yellow flowers. And like most Canary Island endemics, Descurainia arrived onto the islands through a single colonisation event from the continent. And these 7 islands are found across 4 islands. All species are single island endemics, with the exception of Descurainia millifolia, which is found on 3 of the 4 islands. And all species occupy different ecological zones and altitudes. So given the distribution and occupying different ecological zones, this group offers an opportunity to examine the complexities of divergence. And there's little known about the taxonomy of Canary Island Descurainia, so it's important to generate a well-resolved phylogeny. So here I employed 2 phylogenetic methods using a reduced representation sequencing method called genotyping-by-sequencing, or GBS. So firstly, I used the concatenation approach to form a bifurcating tree, which is the most traditional way of using NGS data, by concatenating all the loci to form a supermatrix. However, under this approach, it ignores issues such as hybridization or incomplete lineage sorting, which might alter the topology of the tree. So a second approach was used to generate species networks, which are similar to phylogenies but allow for reticulated evolution, which is the merging of 2 lineages, so i.e. hybridization. And these examine the conflicts between individual genes, which might have different histories due to evolutionary processes, such as hybridization. So since I'm using multiple techniques, it might generate alternating hypotheses. And so I will use a Bayesian modelling approach to find the most likely scenario with the given data. And this also provides additional parameters and allows us to estimate admixture rates between taxa. So the first phylogenetic hypothesis was generated using the concatenation approach. This generated 2 well-supported clades, with clade A restricted to Descurainia preoxiana, Artemisia and millifolia, and clade B being restricted to Descurainia gilva, Gonzalezii, Borgiana, and Emsii. So the second approach was species networks. Species networks were modelled first with no reticulations and with reticulations. The scenario without reticulations, scenario 1, generated a species tree that represents the topology of that of the concatenated approach. However, when we allowed for hybridization and reticulation events, 2 hybridization origins were found. one with Descurainia artemisidoides, and the second with Descurainia gilva and Gonzalezii. So now I've generated two conflicting evolutionary histories, so I need to find out which one's the most likely. So the Bayesian modelling approach found that scenario 2, the scenario where we allowed for reticulations to occur within the tree, it was the most likely with the highest posterior probability. And these results were supported by Abba tests, which indicated that there was introgression occurring between the Gilva Gonzalezii lineage and the Millifolia lineage. So what does this mean in terms of the diversification of Canary Island Descurainia? The numbers next to the reticulations represent admixture rates generated through a Bayesian model. The Gilva Gonzalezii lineage has a similar contribution from their parents, and so this could possibly represent a hybrid speciation event. And ancient admixture events are likely to promote rapid diversification due to the exchange of ecological adaptive traits. And this has provided a possible explanation for the rapid diversification of many lineages. The more recent hybrid event suggests that Descurainia artemisidoides could be an introgressive variant of Descurainia millifolia, as 93% of Descurainia millifolia's genome is shared with Descurainia artemisidoides. So next I tried to put this phylogeny into the context of the species ecology to see whether there was an interaction between ecology and hybridisation in promoting ecological diversification. And traditionally, habitats for Canary Island endemics have been defined through field observations. Here I tried to employ a different approach by extrapolating climate data from over 100 individuals of Descurainia from GIS maps to characterise their climate niches. So I took two climate niche data. The first is the average annual temperature range, and the second is the monthly average precipitation range for each of the 6 of the 7 species. So here we place the 7 species into 4 categories of climate niche based on the average statistical differences between the species and map them onto the tips of phylogeny. Generally, we see there have been 3 ecological shifts, one in warm and dry environments, another to cold and dry environments, and again from dry and wet environments. But yet rarely do we see ecological speciation acting alone, and it's often in combination with other evolutionary processes. So trying to take it into context to the whole tree. Since artemisidoides has been introgressed with millifolia, artemisidoides may have adopted ecological adaptive genes that enabled artemisidoide populations to colonise new ecological habitats. However, since Descurainia artemisidoides and Descurainia millifolia both seem to situate in similar climatic niches, that suggests that this is not the case here. And again, for the Gilva and Gonzalezii lineage, they seem to situate themselves in similar climatic niches from their parental lineages of Descurainia borgiana and Prioxiana. So ecological divergence in relation to hybridisation is weakly supported here, but we're looking into some more extensive ecological characterizations and looking into more local sampling, but at the moment that does not seem to be the case. So the 3 main takeaway points here is that traditional phylogenetic methods could have hidden evolutionary processes, and that in this case hybridisation has been involved, and the reticulated phylogeny creates a new backbone from which to build evolutionary hypotheses from. So for Canary Island Descurainia, there have been 2 hybrid events and 3 ecological shifts. So I'd like to thank some people for helping me with this research, particularly Rachel Ollie and Alfredo for the Descurainia sample, and Barbara Goodson and Bob Jansen for keeping these Descurainia samples, and Oliver White for doing the GBS sampling, and of course my funders as well. So thank you very much for coming to my talk.